So let us look at mutations. In one of the presentations, especially protein synthesis and also cell cycle, I hinted on the fact that it is very important to have these processes properly regulated, because if you have problems with how these processes happen, then you are likely to have disorders, and those disorders, of course, are not good for human health. And that is what mutations actually do. They create alterations in the genetic makeup of an organism. So let's look at how these alterations happen and their impact. What actually causes these alterations? So mutations are sudden alterations in an organism's genetic makeup. That means it happens in the chromosomes or in the genes, because that is where you have the genetic makeup of the organisms. We can also define mutations as a sudden change in an organism's genetic material. This is still to emphasize that the genetic material that you are referring to is basically DNA. Okay, and these changes in the genetic material result into phenotypic alterations, the way how an organism appears, but also the way how an organism appears can also be as a result of alteration in its genetic makeup, that is the genotype. Okay, so the genotype affects the phenotypic appearance. Okay, so these mutations can be inherited. This is to say that they are passed from parents to the offsprings, or they can happen in one's lifetime. That means that after you have been born, some of your cells can undergo mutations. Okay, but if in your life you have what you call mutations in the gametes or in the gamete producing cells, then these mutations can be passed to the offsprings or to the children that you will be having. But the mutations that happen in somatic cells, that is the body cells, these ones cannot be passed to the offspring. So you can't pass them to the children that you will have. Except in situations for organisms that actually have a sexual reproduction, that is when perhaps that can happen. But for sexual reproduction, the passage of these alterations to the offsprings happens if the mutation happens in the gametes. So what are the things that cause these alterations in the genetic makeup? We call them collectively as mutants, substances that cause mutations or that increase the rate of mutations. So they include radiations, and radiations can be of several types. We have ultraviolet radiations, we have X-ray radiations and gamma radiations. And then we have high energy particles, the alpha and beta particles, and some of those examples. And then we have some of these mutants, mutagens actually. So mutants is a different word. Mutants is when you have organisms that have undergone mutation. That means that they are mutated organisms. So they are mutants. So mutagens are those substances that cause mutations. Okay, they increase the rate of mutations. Okay, so the process of cell division, or you can just use the term cell cycle. The process of cell division, or the processes that happen in the cell cycle, need to be tightly regulated such that you don't have errors that happen, because these errors eventually result into alterations in the function of the cells. Okay, and like I've said, These alterations can be passed to the offsprings, or they may not be passed if they happen in somatic cells. Okay, so one of the errors that can happen, for example, during photosynthesis or during cell divisions. In the process of in the cell cycle, we noted that they will have what you call checkpoints. These checkpoints are the ones that check if errors actually, if there are no errors. Okay, if there are errors, those errors have to be repaired before the process continues. Okay, so it is very important that you have DNA replicating accurately, okay, such that you don't have these alterations. So spontaneous changes. This is to emphasize the fact that even sometimes without any external factors, you have alterations in the genetic makeup of an organism. And these are basically spontaneous changes in the DNA. Okay? And this can cause mutations. Sometimes you can have what you call silent mutations. Silent mutations are the ones that you will have changes in the genetic makeup, but you don't have any accompanying phenotypic appearance changes. Those are silent mutations. We can categorize mutations into two: chromosomal and gene mutations. When we talk about chromosomal mutations, we are talking about changes in the structure or number of chromosomes. And of course, remember, we have what we call homologous chromosomes, where you have chromosomes pairing up because they carry the same genetic content. They carry the same genetic content. So if you have, for example, a complete set of chromosomes being removed or added in an organism, then you have mutations. So, and these mutations are called, if you have an organism that has an extra set of chromosomes, then we call that polypody. It's what we refer to that. Usually in animals or humans, it's actually a problem. So you have what you call Down syndrome, where you have an extra chromosome. Okay, that in plants is actually beneficial. Okay, in humans it is not. In plants it can be beneficial in that it can lead to increase the productivity. So that is polypody or pleiopody. Okay, and then the aneuploidy. This is when you have addition of an individual chromosome. The difference between pleiopody and aneuploidy is the fact that with the pleiopody you have an addition or removal of an extra set of chromosome. Actually, the emphasis is on having extra set of chromosome, but here it is just an individual chromosome, just one chromosome, not a set. And when talking about chromosomal mutations, it is very important to know some of the mechanisms that can cause it. So what happens when you have an organism having an additional set of chromosomes? It could be because of failure of the chromosome structure to separate, and that is what you call the non-disjunction. Okay, failure of chromosomes to separate, resulting into one of the daughter cells or offsprings having one extra set of chromosome and then another missing one set, because what is supposed to be shared equally ends up in one of the offsprings. Okay, so Turner syndrome is when there is a missing X chromosome, and then Klinefelter syndrome is when there is an extra X chromosome, and this arises from the failure to separate. Okay, so the different mechanisms of chromosomal changes, especially in numbers, in terms of and especially in structure, you have what you call deletion, where a segment of the chromosome is actually lost, And then we have inversion. It's about the reversal of the segment. Instead of it being the way it's supposed to be normally aligned. Translocation, moving from one part to another, moving one chromosomal segment to another different chromosome. That is translocation. Duplication is having the entire segment of the chromosome being duplicated. Okay, all this can manifest through different physiological ways or developmental ways. You can have issues to do with either delayed development, whether it is cognitive, whether it is achieving certain milestones. All these can happen because of it. Mutations. Gene mutations, we can call them point mutations. They happen when is a change in the structure of a single gene. Still, the mechanisms of deletion, insertion can happen, including substitution. Okay, sickle cell, which is one of the genetic disorders, is actually a point mutation, and it actually follows what we call it follows Mendelian pattern of inheritance. So we'll basically discuss sickle cell disease or anemia under what we call Mendelian disorders. But the points that we have to emphasize here, that it's only a change in a single base, in a single base in the protein that codes for hemoglobin. I mean, in the triple code that codes for hemoglobin, because we know that hemoglobin is a protein with the 4 polypeptide chain. So sickle cell is caused by a single base mutation or substitution. Okay, just a change in one of the bases that codes for hemoglobin, and then you have a problem there. Some mutations can be beneficial. You have also those that are harmful. For example, we have seen in sickle cell, but also many other, the Klinefelter syndrome, the Turner syndrome. All those are complications that happen in humans because of mutations. But not all mutations are actually detrimental or harmful. We have those that are actually beneficial and increasing. If they're beneficial, they have the ability or the capacity to increase an organism's ability to survive and reproduce. And this is basically a basis for evolution. Okay, it's the basis for evolution. So, for example, a mutation that confers resistance to a disease is advantageous and therefore provides what we call a selective advantage, and that promotes evolution. Basically, that means that that organism will be able to pass its deeds to the subsequent generation. Harmful mutations can lead to death. They are lethal. For example, the genetic disorders that I've talked about, sickle cell, Turner syndrome, Klinefelter syndrome, and many others. Silent mutations can be called neutral mutations, in that they have happened, but they do not have an impact on the organism. So that marks the end of this presentation, where we have looked at mutations. It's very important to know the substances that cause these mutations and protect yourself from pain. Frequent exposures to X-rays, gamma rays, and other radiations can actually put you at an increased risk of having mutations. And yet mutations are actually harmful, though we have those that are beneficial. Thank you.